Welcome to HDTV. You're now rocking with your boy. Had to hit the high note on you niggas. Now, scientists finally identified deadly toxin that's been killing birds. Thousands of eagles and other fowl have died from a mysterious condition that attacks their nervous system. Now, after decades of investigation, we know why. The mystery went unsolved. The killer appeared again a few years later in the Carolinas, Georgia, and Texas. In addition to bald eagles, it had started attacking water birds like Canadian geese, coots, and mallard ducks. First, it rendered the birds unable to fly. They stumbled around, their wings drooped, they looked catatonic or paralyzed. Then, in as few as 5 days, they were dead. Now, in a paper published today in Science, an international team of researchers from Germany, the Czech Republic, I forgot how you say it. I think it's Czech Republic, and the United States have finally identified the culprit, a previously unknown neurotoxin called adicothanotoxin, which could be produced by a deadly combination of invasive plants, opportunistic bacteria, and chemical pollution in lakes and reservoirs. To find this new toxin, scientists had to work together like detectives, assessing the crime scene and interrogating suspects. Susan Wild, a professor of aquatic science at the University of Georgia, first began investigating the mystery in 2001 when 17 bald eagles died in Lake Strom Thurmond, a man-made reservoir on the Georgia-South Carolina border. I had seen the eagles' deaths before in the past events, but this one was the reservoir where I had done my dissertation research. She says, it was an interesting mystery, but kind of hit home. That was the reservoir I had worked on and seen a lot of eagles flying over. When Wild had been collecting data for her dissertation in the mid-1990s, there wasn't much vegetation growing in the reservoir. But when she returned a few years later, the lake had been overtaken by an invasive plant called hydrilla, which is easy to grow and had become a popular plant for fish tanks. It's rumored that hydrilla was initially released in the U.S. in the 1950s when it outgrew an aquarium and someone dumped it out into a Florida waterway. Since then, it's become one of the most pernicious aquatic weeds in the country. Thriving in freshwater lakes from Washington to Wisconsin to the Carolinas, Wild began to wonder if the eagles' deaths and the presence of this new plant were related. But Wild had to interrogate all the potential suspects. She started by sampling the water and lake sediment for bacteria. She came up empty-handed, but when she started examining the hydrilla plant's leaves, she found colonies of a previously unknown cyanobacteria. She named it Atocthonos hydrilicola, the eagle killer that grows on hydrilla. Cyanobacteria, also known as blue-green algae, are famous for creating the toxic blooms that poison lakes and seafood. Wild hypothesized that the toxin was produced on the leaves of this plant and then eaten by herbivorous birds swimming around in the lake. When the poison started to work on the bird's nervous system, they became catatonic, easy prey for the bald eagles who migrate south every year to nest. When the eagles ate the infected prey, all the toxins stored in the bird's muscles and stomachs were transferred to the eagles. But to be sure she was pursuing the right suspect, Wild needed to grow some Atocthonos hydrilicola in the lab to find out which toxin it produces. But that's easier said than done. Bacteria are notoriously difficult to cultivate. Plus, she had to culture them in a setting that mimicked the water in the reservoir. It's sort of hard to recreate that environment in the lab, says Wild. The culture keeps getting colonized by other bacteria that grew faster and more readily. We had a lot of trouble with contamination and getting the culture started, she says. That's when Timo Niedermeyer called. Niedermeyer, scientist at Martin Luther University, Halle-Wittenberg in Germany, studies cyanobacteria. And when he stumbled across Wild's work, he was intrigued by this new species. His team had Wild send over a few samples of the colonized hydrilla leaves, and they figured out a way to get the bacteria cultured in the lab. It still grew incredibly slowly. They had to wait 18 months to get enough bacteria to run any tests, but it seemed like they were headed in the right direction. After waiting and waiting, Niedermeyer finally had enough bacteria to run an assay to see what toxins they were producing. They found nothing. This was really frustrating, of course, says Niedermeyer, and we had no idea what to do. By now, they'd already spent a lot of time on a project that wasn't panning out. We worked for 5-6 years without really any results, only cultivating for nothing. He says the scientists had to regroup. They didn't want to be too committed to one theory. You don't want to get so attached to your hypothesis that you can't look honestly at data that says, no, that is wrong, says Wild. But the trick is that negative data doesn't necessarily mean the hypothesis is wrong. It just means that you didn't demonstrate it in that trial, so we did try and try again. This time, Niedermeyer asked Wild to send an entire hydrilla leaf and the stems. Instead of scraping the bacteria off of the leaf, he kept the whole thing intact. He and his team examined it using mass spectrometry, an imaging technique that allows them to see individual molecules on the leaves. Not only did they see the cyanobacteria, but sitting alongside it on the top of the leaf, they also noticed another compound, which contained 5 bromine atoms. Bromine is a chemical element that's highly reactive and isn't usually out and about in the environment. It does appear naturally and it's less reactive negatively. Charged ion form bromide, but even bromide doesn't usually show up in freshwater environments like J. Strom Thurmond Lake. Where they do make frequent appearances are in man-made products. Humans use bromine in sedatives, fuel additives, and to sanitize water. The medium his lab used to grow the cyanobacteria hadn't contained any bromine. Niedermeyer realized that this must be the missing ingredient the cyanobacteria needed to make its deadly toxin. This was like a eureka moment, he says. They added bromide to the mix, and indeed the cyanobacteria produced a toxin. Niedermeyer finally got to call Wild and tell her they found the killer. That was great, he says. Robert Sargent, a program manager for the Georgia Department of Natural Resources, describes the discovery as outstanding news. He particularly excited that the researchers have figured out a way to detect the toxin in the lab. It is just remarkable for ecology, for us getting a better grasp on understanding this process and perhaps being able to control it, he says. He points out that while the eagle deaths are alarming, they are a sign of a much bigger problem. Whenever we see illnesses or deaths of species at the top of the food chain, it's a red flag for the potential health of the environment, he says. After finding the toxin, the research team picked up speed. They isolated the compound containing bromine and confirmed it was present in the dead birds that showed lesions. They looked at the hydrilla plant itself and discovered it's able to enrich bromide from the environment, making it even more available to the cyanobacteria. The concentration of bromide in the plant is much higher than in the water and the sediment where the plant grows, says Niedermeyer. This is kind of intriguing, but we don't know why the plant does it. But in this murder mystery, identifying the culprit isn't quite the same as ending the story. The team still has a lot of questions. Did the cyanobacteria invade with the hydrilla, or was it already in the water? Is the bromine naturally occurring, or could it be coming from man-made sources like coal-fired power plants and flame retardants? Hydrilla is such a persistent pest that people have tried using herbicides like diquat de bromide to kill it off. Could that herbicide be the source of the ingredient that creates this toxin? Wild and Niedermeyer think it's possible. They are also very concerned about whether this neurotoxin could affect humans who eat infected fowl. This could be a real issue, but we don't know that yet, says Niedermeyer. Wild wants to start monitoring it in more locations. Not every lake that has hydrilla has had an AVM outbreak, but there are many where the weed has been treated with herbicide, and they could potentially become toxic in the future. Wild hopes that with more monitoring, scientists can get ahead of possible outbreaks and keep this from spreading even further. Sargent adds that residents can also play a role in efforts to control AVM outbreaks by not dumping aquarium plants into waterways. Boaters can remove aquatic plants from their propellers and holes, and if people see oddly behaving aquatic birds or birds of prey, they can report those sightings to their state wildlife agency. Just managing the outbreaks that have already happened has proven to be tricky. Hydrilla is a tenacious plant. The Army Corps of Engineers has had luck using a grass-eating carp to eat back the weed, but even after being chopped on by fish, it will regrow from tubers buried in the lake's sediment. And even though it grows slowly, Adichthyonos hydrocola is just as hard to get rid of. They simply survive. You can't kill them, says Niedermeyer. He recalls a few cultures and dishes in his lab that had been forgotten and weren't cared for properly. We thought, okay, it's dead, he says, but no, if you just add a little bit of fresh meat in, it starts growing again. Niedermeyer says that's now that they know what they're looking for. Scientists have a better chance of finally stopping the killer once and for all. Now that we are aware of a problem, we can screen for the cyanobacteria. We can monitor the toxin. We can start sampling water bodies for bromine. He says now that we know what we are looking for, we can start finding a solution. So basically, sorry, I know that was long. Basically, I believe it's man-made. And I believe from all the years of dumping illegal stuff and bad stuff into the waters, it damages the ecosystem and it damages the wildlife. So once the thing got onto the plant, once it mixed with that, it made the plant deadly. Whereas if it didn't never touch the plant would have been fine. So when you add certain stuff together, it's like the periodic table. If you add this stuff together, it'll blow up. Like if you add the middle part of the periodic table with the two outside parts of the periodic table, you could create something that is deadly or create something that'll help you. So to me, I think it's a lot of the dumping on the sites and it's a lot of man-made stuff. Like men, like as a society, a global society, we do not care about the globe. We don't care about wildlife. We don't care about none of that stuff. And that's why it will continue to be like this when you have something that just comes up and you're like, man, that is crazy, like how that happened. You know, it just comes out the blue. But you know what I'm saying? Pray for the families. Pray basically. We don't know if it affect humans yet. It might. To me, I think it might, but we don't know yet. Haven't heard anything, but when we do, we'll let you know. Thank you for listening. Like, comment, subscribe, share this. Hit the notification bell to select all to receive upcoming notifications. And if you like to donate to the page, you can cash at me at the word welcome, the number 2, and then HDIITV. Thank you for listening, and we're out. Deezy.